Hello, everyone. My name is Batul. I'm a first year student in a PhD program for chemistry at George Mason University in Virginia. Today, we're covering like a method that we'll be working on it in Dr. Foster Labs for analyzing and characterize the PFAS substance. As we know, PFAS are a human-made substance. They have been under intensive investigation for environmental and public health risk since being detected as a part per million in concentration for the groundwater and surface water. As insulation using firefighter foams, they are considered the main source. The objective is to characterize the isothermal behavior of the individual PFAS by developing a rapid and reliable microsolid phase extraction of the PFAS in water. That would be cost-effective and saving time. Yeah. 24 target substance of the PFAS. They are purchased from Aqua Standards, and they are pre-made mixture in the methanol. The same thing for the internal standard and segregated standard. All water sample that we are sampled in the lab, they've been spiked with the 24 target PFAS in a level 500 microliter and 100 microliter for the internal and segregated standard. The optimization for these method we are working on, it's by condensing and using a 3 ml custom pack SPE cartridges, solid phase extraction, and they are containing 50 milligram of the Stera PFAS wax that layered over 12 milligram of the graphite carbon black sorbents, which is work as a cleanser for our water sample that have been run through the extraction. All the water sample, they are in the volume of 100 ml. They are extracted using the methanol and ammonium hydroxide and eluted with the methanol into 1 ml of auto sample vials. They are polypropylene vials with the polypropylene caps with no need for the solvent reduction or to go through the TurboVap for reduction to 1 ml. During the elution, also we used a syringe filtration. That's a 13 millimeter polypropylene syringe, and with a 0.2 micrometer pore size. They are being used to filter samples to get it ready for the LCMS analysis. The PFAS and quantitative analysis have been done by the LCMS-MS. They are primary analytical instruments that's been purchased from Shimadzu 8050 LC tandem mass spectrometer. 6 to 8 calibration standards are prepared as a dilution of the 24 target internal sin and segregation combined in the methanol. As we see here from the resulted slide, I think I lost it. Oh, sorry. The third slide is a showing of the result from our recovery percentage that have been running after the LCMS analysis and quenching further data. Our PFAS recovery percentage have been ranging between 41% to 91%. And for the segregated recoveries, we are being focusing on the 3 main segregated recoveries that have been ranging between 83% to 103% using this method. This method has been using as also like they are, as I mentioned, they are showing an efficient and a firm result, and they will be cost effective and have been saving the time by not going through going through the TurboVap or trying to have like a solvent reduction.
1 minute.
This method today has been a processing just to use for studying different water samples, which is the field samples like surface water and pool water. Thank you.